Hello, everyone. Greetings from New York City. My name is Viviana Simon. I'm a professor of microbiology and medicine at the Icahn School of Medicine here in New York City. I look forward to sharing some of our recent findings regarding SARS-CoV-2 surveillance in New York City. As you know, New York is the largest city in the United States with over 8.5 million inhabitants. We also welcome millions of domestic and international visitors every year. Outbreaks of pandemic pathogens have threatened and will continue to threaten world health. As an international hub, New York City is a very likely entry point for pathogens with pandemic potential. The Mount Sinai Health System is the largest healthcare provider in the city, with 8 hospitals and many ambulatory sites. Every year during the winter season, we see over 1,000 patients seeking care with influenza-like illnesses in our hospitals. When we heard about this new coronavirus emerging in China and in Europe, we rapidly retooled our pathogen surveillance program to accommodate this new virus. The pathogen surveillance program is institutional-wide program that we set up a couple of years ago in order to map the spread and evolution of pathogens in New York City. So we collect clinical specimen. For example, we have nasal pharyngeal swabs from over 5,000 SARS-CoV-2 infected patients in our repository. We also link this large biobanking efforts with electronic databases that allows us to retrieve patient demographics, medication, and we also provide sequencing of the viral pathogens. The next slide shows you some of the actors that actually do the work. Here we see Dr. Sordillo, who leads the clinical microbiology lab, Dr. Harm van Bakkel, who is in charge of the sequencing efforts. Here, my lab is shown here. We do the biobanking and molecular diagnostics for the viruses. And here you see Dr. Kramer, who oversees the antibody work, which I will have no time to talk about today. Coronavirus itself really needs no introduction since we all have become experts over the past months. Most of my talk will be about tracking the initial spread of SARS-CoV-2 in New York City. I will end at talking about continued evolution of SARS-CoV-2 and how we can use genomics to track new transmissions. The first cases of corona were identified on February 29. The first case was actually seen at Mount Sinai Hospital, and subsequently rapidly additional cases were identified. Using the pathogen surveillance program, we were able to secure the samples and rapidly sequence the viruses that cause disease in those patients. And we shared, within weeks, we were able to share the sequences with the scientific community. And this slide shows you how dramatic the case numbers were in New York City. As comparison, I'm showing you here cases with influenza A and B. This was a severe influenza season here in New York City. Yet, when you compare to SARS-CoV-2 infections that we saw in March and April of the year, this is really no comparison. So SARS-CoV-2 is nothing like influenza. The peak of the first wave in New York City was reached early April, and then the case numbers slowly but surely came down, so that here up in the summer we had very few cases. We see approximately now 5 to 10 infections in the entire hospital system. The first 2 cases, as mentioned, had a travel history. New York 1 had a travel history to the Middle East, while New York 2 had a travel history to Spain. So in both cases, those patients acquired the infection outside of the United States. We were wondering if this was a common pattern or if there was other sources for the infection in New York City. So we started to sequence the first 84 genomes from patients seen at Mount Sinai here between February 29 and March 18. And we were able to assemble complete genomes of those cases. And this phylogenetic tree shows you that there were multiple independent introductions into New York City. To our surprise, those introductions came mostly from Europe, and on some occasions also from North America. We had not found a single case of direct introduction from Asia. Another surprise was this cluster I'm showing here with the arrow, which suggests that there was untracked community transmission in New York City. Here by many cases sharing a very similar viral clade. By looking at the geographic distribution of the COVID cases for which we sequenced virus, we see an accumulation here of cases in Manhattan, close to the main campus of Mount Sinai Hospital, as well as Brooklyn, where there is another hospital. The clade distribution across all those sites was very comparable. So there is no evidence for local transmission supported by this data. So it seems there is diverse viruses in all neighborhoods of New York City. By looking at the most common ancestor of the strains we see, we can infer that there must have been untracked transmission to explain those cases here. The largest cluster has 69 individual cases, and we predict that the introduction occurred in February. So there is untracked cases back in February. This is a phylogenetic tree, which here suggests what happened in this window. Since we have no documented cases here, we need to take advantage of phylogenetic tree reconstructions, which I'm showing you here in the next slide. This is a blow up of the sequences that are found in this large cluster of community transmission. And you can see that the cases we see at Mount Sinai are also comprising a case which from a person in Westchester, which had the first documented community spread early March in New York City. So this really suggests that there was untracked transmission in the early phases of the outbreak in New York City. And we have continued our surveillance effort, and as this timed tree shows you from January to July, the sequences we have generated. By now, we have over 700 complete SARS-CoV-2 genomes. In red are the viruses derived from patients seeking care at the Mount Sinai Health System. And in yellow are the strains derived from patients seeking care at other medical centers of the city. And you can see that there is a large diversity in the viruses obtained from patients seeking care at New York City hospitals. And this diversity is maintained as time progresses, suggesting that there is continued introduction and circulation in the city. So that it's not a single cluster, but there are continued introductions into the city, despite our prevention efforts. This shows you just the absolute numbers of genomes. We have sequenced over 300 genomes in March, and as time goes by, we are continuing to sequence. Our goal is to have 100 sequences per month as the pandemic continues. You can see here that the clade distribution evolves over time. Again, we need to be careful because our case numbers here are still different. But you can see here in July, we basically have 3 clades that dominate. that are 20A, 20B, and 20C. The big open question is, does evolution of SARS-CoV-2 change the pathogenicity of the virus? And one of the mutations that has been looked at is a mutation in the spike protein at position 614. And this time tree shows you basically that there are two flavors here, with in yellow the mutant viruses, and you can see that all of the contemporary viruses that we see in June and July in New York City do carry this new mutation in spike. So this graph shows now the dynamic changes of clades at a global scale, and you can see those are the 10 most common clades that we see. And you can appreciate how things change over time. We are Marta Luxer, who has developed Flu Predict, is adapting the platform to follow the dynamic changes for SARS-CoV-2 with the ultimate goal of predicting fitness and outcomes. So this is work in progress, and we are doing similar analysis now for the viruses seen in New York City. Here, this is we are starting to link sequences with outcomes. And here we have in this graph the sequences for the people that have been discharged from the hospital in blue, and those that are deceased in green. And you can see that the green dots are found across all clades, suggesting that there is no single clade that is more pathogenic or deadly than the others. Here we need to sequence more, but this is what our current data suggests. Here is New York City. This shows you the case numbers for the 5 hospitals from Mount Sinai. And you can see that in all locations, the numbers came down dramatically, where now here we have little spikes, which creates the opportunity for contact tracing and genomic surveillance in order to identify potential clusters early and intervene. This is an example of 2 co-workers that presented with COVID-19 symptoms 3 days apart. And we obtained the nasal swabs and successfully sequenced the viruses. Here we have case A and case B, here shown on the phylogenetic tree. And when we looked at the individual variants found in those samples, we found that those 2 viruses were encoded a number of unique signature mutations, including mutations in spike found in case B, which when we look back at the minority variants in case A were also seen. So this is a possible example of how the virus adapts when it gets transmitted from one person to the other. Ongoing experiments will tell us if those mutations are increased with, are associated with increased fitness or antibody escape. So to summarize, SARS-CoV-2 is a case of the largest non-influenza pandemic of the century. New York City was one of the early epicenters of the outbreak in the United States, with over 230,000 infected people. By studying the sequences of the early viruses found in New York City, we've obtained evidence for multiple independent introductions with local forward transmission and from undetected early introductions. As we continue our surveillance efforts, we focus on the current patterns that suggest ongoing small-scale domestic introductions. We also are in the process of studying the impact of public health measures on the transmission and interaction with other respiratory viruses, especially influenza, as the season starts. And we are really looking for nosocomial transmission within the hospital system. With this, I want to thank all my collaborators, especially the Van Bakel Lab, the Clinical Micro Lab, the Luxer Lab, and all my colleagues that makes this possible. I'm also grateful to the support from the leadership and to the support of Dr. Palesi, the chairman of the department. And I thank you for your attention.